I want you to understand the basic types of research that are done into the human microbiota so that you can understand where we're at and what the limitations are of studying it. So there are really two types of studies, and I'm going to show you some examples of them. The first type is animal experiments. There are amazing experiments done with mice where researchers can control which bacteria grow in them, and so they've seen just unbelievable effects of the gut bacteria on mice. For example, gut bacteria in mice have a huge impact on the mice central nervous system and their overall energy metabolism. They can make a mouse morbidly obese just by giving it a certain type of bacteria that will more efficiently extract energy from its food. And they can do the opposite. They can give it bacteria that will make it skinny. These have never been done in humans. Obviously, it would be really cool if we could control or how fat or skinny we were using bacteria we could eat, but there's nothing like that in humans. What we do in humans is we can't do those experiments, but we can do correlational observational studies where we describe the microbial community structure in a person, and then we correlate that to other health data we get. So let me show you an example. This is going to be an example of an animal study. And in this, what they did is they wanted to see if antibiotics could cause dysbiosis and if they could break colonization resistance. So what they did is they got mice and they fed them vancomycin-resistant enterococcus. Don't worry about what it is, but it's a pathogen that is a gut pathogen. Untreated mice, so right here, were not colonized by those pathogens. So they ate the pathogens, but when the researchers looked in their guts later, in the small intestine or the cecum, those pathogens were not detected in the small intestine or cecum. Now basically what this is showing us is this level here is the smallest they could have detected, and they didn't detect it. So they know the actual number was somewhere below this. And this is a log transformed scale. So every number here, think of it as 10 to the first power, 10 to the second power, 10 to the third power, fourth power, fifth power, sixth power. So this is an exponential scale, as is this. Right. So other mice, so that's the control mice. Other mice, they gave antibiotics to see, okay, if we disrupt their bacterial community with antibiotics, what's going to happen? So pause the video and think about before you pause, think about, right after antibiotics were given, how many of these pathogens would you expect to find if they were fed to the mice after the antibiotics? So the mice eat the antibiotics, and then they eat the pathogen. How much of the pathogen do you expect to see? Pause the video, guess. Well, here's what they actually saw. When the gut community was really reduced because of the antibiotics, it had no ability to prevent these pathogens from growing in the gut. But the real question is, what's going to happen 2 weeks later? 2 weeks after the antibiotics, the bacteria in the gut have regrown. Those mice have robust communities of microorganisms in their guts. So, what are you going to see? If those mice eat the pathogen, will it be detected? Won't it be detected? Pause the video. Guess. This is what they found. Even a month later, and that's a long time for a mouse, the mice did not regain their powerful colonization resistance. So their microbiota, when it grew back altered in a way that was not protective. So the antibiotics caused dysbiosis, and they broke colonization resistance. So that's that. This is a different study. This is a human correlative study, I guess, where they got stool samples from people, and you're going to see these pie charts. So this is one person's stool sample, and the different slices of the pie represent how much of the bacteria are from this phylum, or this family, or this phylum, or this phylum, or this phylum. So first I want you to look through this and see, could you predict what a person's... So this is 5 different healthy volunteers. And you can see some of the challenge of human microbiota research here if you try to describe what does a healthy microbiota look like. So pause the video. Go for it. Describe healthy microbiota based on these 5 samples. There aren't really any patterns that always hold, other than there are some firmicutes in these samples. But this healthy person, the Bacteroidetes are common in 4 of the samples, but not this one. So the next thing we're going to see is stool samples from intensive care unit patients. And on this side, it's patients who died. The black dot is when they died. The numbers are days after they were admitted, I think, to the hospital. I don't remember. But 70 days after admission, this person died. And then on this side, it's people who were discharged from the hospital. So they survived, and they were discharged, these green dots. So looking at people who were sick and eventually perished, or people who were sick and eventually at least left the hospital, can you see a difference? Can you see any sign of a unhealthy microbiota? Go ahead and pause the video, look around, see what you can find. So again, there's no obvious trend that holds up in every case that I've ever seen from these data. One thing we noticed, the authors point this out and it's pretty clear, is that these Proteobacteria show up in some of these, in large quantities in some of these intensive care patients, but not really in the healthy patients. Proteobacteria are facultative anaerobes like E. coli, and they really shouldn't be able to compete against obligate anaerobes like the Bacteroidetes in the gut. They really don't have, they're not specialized for growing in the gut the way some of those other bacteria, like the firmicutes. Clostridium, the really amazing obligate anaerobe fermenters, are in the firmicutes group. These 2 really are what you expect to do well in the gut. Proteobacteria, no. We could think of them as weeds that grow in the forest when it is disrupted. There are other things we could see. And so basically, if you want to come up with some answer about what's the difference between a diseased person's gut and a healthy person's gut, really what you need is thousands of diseased people and thousands of healthy people and really high-power statistical software that can answer these questions for you, because no human is ever going to find all these patterns. And we haven't ever found any interesting patterns. Anyway, so that's another study. This one is a mouse study. And what you're looking at here are villi in the small intestine of a mouse. And the blue, well, this is fluorescent microscopy. And the blue is a fluorescent stain on the DNA of the epithelial cells in the villi. And the green is a stain that stains bacterial cells. Now, wild type means normal, healthy mouse, not a mutant. This is a mutant mouse. It is missing one protein. It's missing the ability to make one protein. And the protein it's missing is an antibacterial protein. So in the healthy mouse, there's normally a separation between the microbiota and the actual host cells. And you better believe you'd find the same thing in a human colon. We do not expect these bacteria to actually touch our cells very much. We have a thick, thick mucus layer that prevents that, and any bacteria that can chew through the mucus are pathogens, and we have immune responses against them. So without that antibacterial protein, a lot of these bacteria get through the mucus, and they're all over the villi, and the villi are swollen and misshapen. So one essential thing for controlling the microbiota is being able to kill a lot of bacteria whenever we want to. Sorry, I forgot to mention you can skip this one if you want to, and you can also skip this next one. This one is wild, though. This is a really important thing to think about. So I'm going to walk you through it. A, B, and C are people who donated stool samples at multiple times. So we're looking at the bacteria in their guts. Each of these numbers corresponds to a different type of bacteria in the gut, and the color, whether it's bright yellow or black or anywhere in between, is how abundant those were. So if you look at 10, that's some species, I don't know which one, and in some of these samples it's bright yellow, in others it's not. Here it was abundant in this sample. It was not abundant in this sample. And so there are hundreds of different bacterial species in each sample. And if you look very carefully, you can see that this person gave 1, 2, 3, 4, 5 stool samples. So there are 5 distinct stacks of these different colored bars for this person. 5 for this person and more for this person. So now let's get to the experiment they did. These were people who were hospitalized and given ciprofloxacin. And they were given ciprofloxacin antibiotic. When they were given that, that's these blue bars. And the question the researchers were asking was, does the microbiota come back to normal after ciprofloxacin is given? And if it did, we would see the same pattern on the right side of this very disrupted pattern as we would on the left side. And a lot of microbiologists were encouraged by this because it does look very much like the microbiota come back to normal after Cipro was given. But we know this isn't the case for other antibiotics. So this is the thing we look at. Okay, that's enough research for now. Next, we'll talk about practical information.